I'm just going to present our submissions on the Biogen task from the Webis Group or the Friedrich Schiller University in Jena. So basically, I just want to quickly recall the motivation of this shared task. So we often have situations like this where we ask ourselves some biomedical question. For example, here we want to know what the active ingredients are from some medication. And in this case, probably most of us would just go and use ChatGPT and ask ChatGPT for an answer. And the issue with ChatGPT in this case is that it does not give background information, important background like the dosage, and doesn't cite sources, even though it does give the correct ingredients after all. So another option would be to use your favorite search engine and just click on the first result. So you will likely get very relevant information. It does list the correct dosage and it is a good source to cite, but it takes a bit longer. And for the Biogen scenario, we want to streamline this process so that we can use generative models and have the flexibility and verifiability of web search. And also, just for fun, consider this very basic baseline where you just look at the physical package of the medication and you get all the information you needed from a good source. I think this is the fastest method to get an answer on this particular question. So mainly we want to do medical work, and we thought that this is very similar to medical question answering, and we participated at the CLE BioAsk lab earlier this year. And as BioAsk uses a very similar QR style evaluation setup, we wonder why not just improve our systems and resubmit here at TREC Biogen. So the most important difference maybe to BioAsk is that BioAsk does not require us to cite references, but still we give references along the generated answer. The good thing, of course, is that we have very large training data, over 5,000 questions that we could use. And for each of these questions, we get a summary answer, somewhat similar to the answer type expected for Biogen. We also get some ground truth exact answer. That is, for example, a yes or no, if the question was a yes, no question, or some fact or a list of entities. And we get relevant documents, in this case also abstracts from PubMed and snippets or passages from these relevant abstracts. Interestingly, also BioAsk shifts towards a more RAG style evaluation. So they had in this year, they had some tasks that also required you to do a retrieval and generation in one step. Okay, so our basic RAG pipeline does look like this. We have 4 stages, and in these 4 stages, we first retrieve some relevant documents, in our case, medical articles from PubMed. Then we would extract and re-rank some passages from these abstracts, as the medical abstracts tend to be very long, and rank these passages by relevance. Now these, the top ranked or most relevant passages can then be used as context for some LLM to generate first a summary type answer with citations, but also to generate a sort of exact answer, as I said, yes or no, or a fact or a list of facts that we could maybe use later on to refine our retrieval. And then we could also think about how we combine these generation focused and retrieval focused components into a RAG pipeline. So there are several augmentation patterns that we could use here. Let's talk a bit about the document retrieval. Here we indexed the full PubMed baseline from 2024, but also filtered on the allowed list of PubMed IDs that were given by the TREC Biogen organizers. We indexed all the metadata along with the abstract text, title, and full text. So we used the same API that is used in the unpaywall browser extension to find open access full text for about 10% of the PubMed. And then we would just match the query or narrative or question on the abstracts, the titles, the mesh terms annotated for the PubMed abstracts, and or maybe a previous answer that we got from some previous generation step. We also experimented with some filtering. So, for example, we disallowed articles that did not contain a title or abstract, which seems to be quite common in PubMed. And we also filtered based on the publication type, where we disallowed certain non-peer-reviewed types of publications. So basically, no for the document retrieval, we just use BM25 and some filters. So is BM25 still any good after 30 years? I think yes, but let's see the results then. Okay, so the passage extraction is also very simple for our approach. We used a rule-based approach where we just split the abstract into sentences and then use passages which are basically just n-grams of consecutive sentences from the abstract, up to 3 sentences. We also use the full title as one additional passage, and then first we rank these passages pointwise using either Monody5, TASB, Enki, or Colbert. And then again, we rank some of these passages pairwise using a duo T5 model. Now for the generation, we did not want to manually engineer prompts, so we used the DSPy framework that allows sort of programming LLMs. So you give DSPy a signature as in the form of a Python class, some short explanation of what the expected output would be, and then DSPy will template some prompts and can also tune few-shot examples. With DSPy, we used 2 LLMs, Mistral 7B and GPT-4 Mini. Then gave the models the top K passages and maybe some previous answer for refinement as the inputs. And then, yeah, DSPy would basically then do all the prompting for us. And we could also tune DSPy based on the bioask data to include some good few-shot examples. Here, the main question would probably be if we need manual prompting at all, or could we just rely on DSPy doing all the prompting for us? Last but not least, we looked at some different augmentation patterns. We saw some earlier, in the earlier talks, we already heard some interesting patterns there. The basic idea is that the easiest pipeline you could think of is just use no augmentation. So you retrieve and generate completely separately You just retrieve documents for the for the document retrieval part and generate an answer using no context at all. We can also do RAG. So the retrieval is used to sort of guide the answer generation of the LLM by passing those relevant passages or documents to the LLM as context. But we can also do the other way around. If we have a generated answer from some LLM, we can use retrieval to verify this generated answer, but also to improve the retrieval itself in a query expansion fashion. And then I don't think there is a limit. You can also do some recurrent augmentation patterns. So you could probably, for example, just first retrieve, then generate, and then again retrieve, or again generate, as long as you are willing to do so, and until you are happy with the generated answer. And for these recurrent patterns, we experimented with two types of recurrent augmentation. So the first approach would basically just be that we augment the generation part and the retrieval part independently. So for the generation, we would, for example, just generate a first iteration of the answer, then retrieve some documents and use these retrieved documents to refine the answer. Similarly, for retrieval, we could do some initial retrieval, generate some answer based on these retrieved results, and then refine our retrieval using query expansion. But we could also do like what we dubbed cross-augmentation. So we could use the same generation and retrieval modules and just switch out the outputs of those two modules for a number of steps. So the generation output from the first one is passed to the second step for the retrieval and so on. Yeah, for our submitted ones, we just made every decision in this pipeline a hyperparameter and tuned those hyperparameters with Optuna. Also tuning the few-shot examples with DSPy. So for training, we just sampled 10 random Q&A pairs from the BioAsk 2024 training set and looked at the recall at 1000 for the retrieval part and NDCG. And for the generation part, we looked at Rouge 1 and Rouge L as the objectives for the hyperparameter tuning. Then for each of the two considered LLMs, we ran 100 initial trials using these 4 objectives. And then for the top 10 trials per LLM, we re-evaluated with 2 additional objectives that are more costly to evaluate because we use LLM-based measures, faithfulness and answer relevance from the Vargas toolkit. And from these ranked trials, we then selected up to 5 best parameter choices per each LLM for our TREC submissions. Afterwards, we just semi-automatically post-processed the outputs to fix some sentence splitting issues. I think there was something in the chat also regarding this, and we just wanted to fix it before we submitted the ones. So we have 7 ones in total, 4 Mistral-based and 3 GPT-based. I don't want to bore you with many numbers, but just give you some of our findings. So we found that consistently GPT-4 Mini outperforms Mistral 7B in almost every metric. We saw that having some retrieved context seems to confuse GPT-4o. Actually, the highest accuracy we of our ones was without using retrieval at all. And yeah, Mistral most commonly misses aspects of the answer, so the answers are kind of incomplete. Yeah, but there's no clear winner of the augmentation patterns we considered. So every pattern was in our parameter optimization, and all were fairly similar from the results. Our ones had kind of a bad recall, both on the answer level, but also on the document level. The only caveat here being that the evaluation of the document recall could be biased in the sense that only documents that were referenced in the text were allowed to be submitted as documents. So maybe our models would have retrieved other documents that are not cited in our generated outputs. To summarize, we got kind of mixed results due to relying on these random parameter choices from Optuna. So there's no clear winner of our ones. We did not have a common baseline, which is something we can improve, of course, in the next iteration. We did not have a evaluation of the retrieval part in isolation. So the retrieval evaluation always depends on which text was generated. So in the future, I would imagine that we just do some more comprehensive parameter tuning, considering more trials to get a better tuning. And also one question I want to bring into the discussion is whether we could maybe just reuse the extracted passages directly for nugget-based evaluation. So this could be something that the task organizers could just use as we generated anyway, and maybe they are close enough to nuggets so that we can use it for evaluation. Of course, our code is online on GitHub. And then with that, thank you for listening. And I guess I see many of you next year at Clay BioAsk or TREC Biogen.
Any questions?
We have a virtual, Mr. Udo Kirschwitz. Please on mic.
No, I was just applauding.
I'm sorry, I saw your hand raised. Thank you there.
I have a question. You said that doing retrieval kind of didn't help you, and you just generated the response using the language model.
Yeah, it seemed like at least Mistral struggled to cite the documents that we gave as references. So maybe we had, for example, we may have had 10 relevant documents in the context for the LLM, and it just cited maybe 2 of these. So this could be an issue.
So a lot of the information from PubMed, I guess, has already been used as training data for these models. There's a strong bias there.
Yeah, of course. Yeah.
Got it. Thanks.
Any other questions? Okay, if not, then we will go for break and we will start at 2:50 p.m. again. Thank you.